Hello everyone, this is Dr. Fox, and let's talk about the internal features of the heart. In this video, we'll describe the features common between atria and ventricles, as well as those particular to each chamber of the heart. We'll describe the aortic sinuses and their significance, as well as how heart valves are structured and work. So let's begin our study of the internal features with the right atrium. It seems as logical a place as any to begin. The right atrium receives blood from the systemic loop through two major conduits, the superior vena cava and the inferior vena cava. It also receives blood from coronary circulation, or from the coronary sinus that is from the myocardium of the heart. Now, it's interesting that the blood is received in an area of the right atrium called the sinus venerum. That is basically the space of veins. And the hallmark of this space is that it is delightfully smooth-walled. In fact, the sinus venerum is the posterior aspect of the right atrium, and it is separated from the anterior region by a ridge of myocardium called the crista terminalis. Now here we can see the right atrium has been incised and reflected over. Anterior to that crista terminalis, we see the pectinate muscle, and that's going to be inside the auricle of the right atrium. Now, there is also a sulcus terminalis on the superficial portion of the heart that aligns with that crista terminalis. Why this is also significant is if we follow the crista terminalis all the way to its most superior part of the heart, round about a place of the interatrial septum, the wall between atria, we come across a very interesting collection of cardiac myocytes. Now, this region is called the sinoatrial or SA node. You may also hear it referred to as the sinoatrial node. A rose by any other name would still smell as sweet. This is the pacemaker region for the heart, and it is upon this region that the extrinsic cardiac plexuses, those autonomic plexuses, will work to help regulate heart rate. In addition, we have an interatrial septum here, and that interatrial septum separates the right from the left atrium. So we'll also see this again in the left atrium. One of the more significant portions of this septum is a region called the fossa ovalis. Fossa is an indentation or depression, and ovalis because it's window-shaped. This is a remnant of the foramen ovale, which is present in a fetus. The foramen ovale represents a shortcut for fetal circulation. Oxygen-rich blood ascends from the placenta ultimately up through the IVC, and rather than continue on through the right side of the heart, which would lead to pulmonary circulation, in the fetus, the lungs aren't carrying the burden of oxygenation. Rather, oxygen is coming from maternal blood, so that richly oxygenated blood is heading through that oval window, that foramen ovale, into the left atrium so that it may shortcut fetal pulmonary circulation. At birth, there are two septa that snap shut, and when they snap shut, the foramen ovale becomes the fossa ovalis. In some individuals, a small patency may continue, and that's called a patent foramen ovale, or a PFO. Very small ones are typically subclinical, completely asymptomatic. Very large ones may require intervention. Now, there's also a septum between the atrium and the ventricle, and it's within the tricuspid valve is located. But where the atrioventricular septum meet in the interatrial septum, we have another specialized collection of cardiomyocytes called the atrioventricular, or AV node for short. So the SA node is the pacemaker region, and that is going to control the rate of depolarization of the AV node. And just adjacent to that, we have the opening for the coronary sinus. The coronary sinus is the space into which most of the cardiac veins of the greater system are going to drain. So the blood supply of the heart is returned back through venous channels through this coronary sinus and into the right atrium. Blood will then flow through that tricuspid valve into the right ventricle. The tricuspid valve is named that way because it has three cusps to it. There's an anterior, a posterior, and a septal cusp. And each of those cusps are anchored via chordae tendineae, tendinous cords, which we can see here, to extensions of myocardia called papillary muscles. And just like there were anterior, posterior, and septal cusps, there are anterior, posterior, and septal papillary muscles. And the anterior tends to be the largest, as we can see right there. In addition to this, there are going to be large, meaty beams. So these would be extensions of myocardium called trabeculae carni. You can see that written out right there, applied to the left ventricle. But these are all throughout the right ventricle as well. A particularly specialized trabecula carni is something called the septomarginal trabecula. So the septomarginal trabecula is a specialized bit of myocardium that runs from the interventricular septum to the base of the anterior papillary muscle. And this was first described by Da Vinci, and it was thought to provide structural strength to this, but we now know that it is a shortcut for the right bundle branch of the conductive system. Sometimes this septomarginal trabecula is called the moderator band. So here we have that trabeculae carni, or the meaty beams, all nicely spelled out for us. We can see this interventricular septum there. And if we note the flow of blood, we'll see that it goes through the tricuspid valve down, and with ventricular systole, it's pumped in an ascending way up through a region of the right ventricle called the conus arteriosus that focuses that blood on the semilunar pulmonary valve. The pulmonary valve separates the right ventricle from the pulmonary trunk, keeping in mind that this pulmonary trunk is conducting very deoxygenated blood that's come from the systemic loop that we're sending out to the lungs. So there is the pulmonary trunk, and then there is the left pulmonary artery, and the right pulmonary artery tucks away underneath the arch of the aorta there. So these pulmonary arteries are vessels that carry blood away from the heart, are conducting some of the most oxygenated blood in the body. And we're so used to arteries conducting oxygenated blood. Now, that blood goes to the lungs, it exchanges its carbon dioxide so that the body can expel it, it picks up oxygen, and it returns to the heart via pulmonary veins. There are 4 of these, and the left atrium is situated posteriorly towards the posterior wall of the base of the heart. And we can see that incised here, and with this incised and reflected, we can see that a good portion of that left atrium is also smooth-walled posteriorly, and we can see some pectinate muscle, just barely there, anteriorly in association with the auricle of the left atrium. Now, just as there was an interatrial septum that we could see from the right atrium, we can also see it from the left atrium, and we can see that fossa ovalis. Blood coming back from the lungs will then travel through this bicuspid valve, which is part of the left ventricle. So let's start there. The bicuspid valve is called that because it has two cusps, and it resembles a bishop's mitre or hat. So sometimes we call this the mitral valve. And just like on the right ventricle, we have chordae tendineae, and we have papillary muscles. There are two valves, or two cusps for the bicuspid valve, an anterior and a posterior, and so we have anterior and posterior papillary muscles as well, which we can see there with those chordae tendineae anchored to them. Just like on the right side, we also have trabeculae carneae, or those meaty beams that are arranged to maximize structural force during ventricular systole. There is an interventricular septum that can also be seen and palpated. Now, if you're looking at each side of the ventricles, you might notice that the myocardium of the left is thicker than that of the right. And that's because the left side of the heart needs to be stronger to pump out blood to the entirety of the body through that systemic loop, whereas the right side of the heart need only pump blood out to the lungs. The left ventricle is going to accomplish the role of pumping blood to the systemic loop by pushing it through the semilunar aortic valve, and that aortic valve feeds into the ascending aorta, which is the proximal most part of the aorta. Let's take a look at those valves up close and personal. Here we can see all four of the valves. The atria have been stripped away, and so we're looking at a supero-posterior view of all of the heart valves. And so this is the pulmonary trunk, and this is the aortic valve. Here is the tricuspid valve, and then here is the bicuspid valve. All right, so let's look at the particulars of the aortic valve. So the semilunar valves get their name because each of their cusps looks like a semi-moon. And so we have a right cusp and a left cusp and a posterior cusp. So right, left, and posterior. And beyond these cusps, we have spaces. So we have a right aortic sinus, a left aortic sinus, and a non-coronary sinus. Sometimes we call these the sinuses of Valsalva, is the eponym. The reason why they're significant, we can see here, is that we have coronary arteries arising from them. So this is the right coronary artery, and over here we have the left coronary artery. So coronary arteries arise from the aortic sinuses, the right coronary artery from the right sinus, the left coronary artery from the left sinus, and the space above the posterior cusp is called the non-coronary sinus because no coronary arteries arise from it. Let's take a look at how these semilunar valves work. It's really all about increasing pressure in the ascending aorta and the pulmonary trunk. So as pressure increases after it's been expelled from the ventricles, there's going to be a differential. Greater pressure in those arteries, lower pressure in the ventricles. Now what that's going to do is favor the movement of blood back towards those ventricles. But what happens is, because of how these cusps are structured, each is like a little scoop, and as blood begins to come back, it catches on these valves and it reapproximates them. It pulls them together. So higher pressure in the ascending aorta and pulmonary trunk close the semilunar valves. Thus, during systole, when ventricular pressure is high, the semilunar valves are open. During diastole, when pressure is higher in the arteries versus the ventricles, those valves are closed. Let's look at action of the atrioventricular valves. This is all based on ventricular pressure. When ventricular pressure exceeds pressure in the atria before them, the cusps of the valves are going to catch blood and approximate themselves. A lot of people think that what happens is the papillary muscles contract and that pulls on the chordae tendineae and that closes the cusps, but it's more like a parachute action. Those chordae tendineae do put tension on the cusps. The papillary muscles do help provide that tension, but it's pulling against the pressure of the blood as it's exerted on those valves. And it's that pressure that brings the valves together, even though the chordae tendineae are helping that. But their true function is to prevent valve prolapse so that it would blow open backwards and allow blood to flow through. So as a consequence of this, during diastole, the AV valves are wide open. And that makes a lot of sense because most of ventricular filling during diastole, about 3/4 of it, occurs during diastole. It's only the last 1/4 of it that happens during atrial systole. And then during ventricular systole, those AV valves are completely closed. So let's assess the knowledge. What structures anchor atrioventricular cusps to papillary muscles? A. Chordae tendineae? Yep. B. Crista terminalis? Nope. C. Pectinate muscles? Well, not. Those are in the atria, so not so much at all. Septomarginal trabecula? Nope. Trabeculae carni? Nope. Correct answer is A. Chordae tendineae. Thank you so much for your time, and I look forward to talking to you in the next video. Bye now.